Hello, this is Zhou from Northeastern University. Today, I will discuss the characterization of AAV. In particular, I will focus on the deamidation of asparagine and a summarization that lead to isoaspartic acid. This is a very complicated process, so I will discuss the challenge of analysis and particularly artifacts and also opportunities for capsid characterization. Let me turn my laser pointer here. So I will briefly talk about the impact of deamidation on AAV, and then I will spend some time on the chemistry of deamidation and isosummarization, particularly the mechanisms and the factors affect these processes. Then I will move on to the artifacts of these analysis. I think this is an area that has not been emphasized enough. And also share some good news, how we monitor the artifacts and how to control them. And then the later part of my talk, I will have a little more forward thinking and share some ideas of isoaspartic acid, which is a beta peptide, very much change of protein conformation, how it is formed, and what's the impact. And also then bring to some discussion about intact protein analysis. I think this is particularly important for viral capsid and how we do that, perhaps from a chemoenzymatic labeling method. And then a little bit about how to track each particular step and optimize the process to minimize and avoid deamidation. And finally, very briefly, share with you some ideas about biotransformation, essentially what happened in vivo for deamidation and other PTMs. So the first, I will talk about the chemistry of deamidation for people who are not familiar with this process. So this is asparagine residue, which has a amide bond on the side chain, and the amide bond on the backbone, there's a lone pi electron can attack the amide and form a succinimide intermediate. This is succinimide intermediate can hydrolyze at this bond and form carboxylic acid. This will be aspartic acid. The other bond in succinimide can also hydrolyze. That will form a isoaspartic acid. I will talk about isoaspartic acid in a minute. So these are very common protein modifications. Since they're spontaneous process, means they don't need any enzymes to occur. And as long as there's water, this deamidation or summarization process occurs. So basically can happen under many conditions. And they are ubiquitous because these residues are common, also means under many conditions can be formed. So just illustrated here is the deamidation rate, shows over here. That depends on many factors, can be as fast as 1 day. That depends on the primary sequence, depends on the higher order structure and also pH dependence. In a peptide without higher order structure, asparagine followed by a small glycine deamidate as fast as 1 day, the half-life, followed by a serine residue or histidine deamidate about 10 days. And when they followed by a secondary amine proline, there's no deamidation. To help my students, others to memorize this trend, again, in the peptide, NG stands for not good. It deamidates very fast. NS is not sure. NH is not happy, means they deamidate in a relatively slow but considerable degrees when in a peptide or no higher order structure. So here again shows the peptide sequence dependence. Again, I want to emphasize this depends very much on the structure. The other one thing is to emphasize here, shows here the structure change. If a protein is folded and the higher order structure may stabilize the protein from deamidation or summarization. But if the conformation change illustrated here, then deamidation or other events may happen. So this is very important to monitor the conditions and the conformation change during all these processes. The other major factor for deamidation and the related process are pH. So you can see typically deamidation occurs at a higher pH, and also maybe also at low pH. So typically at mildly acidic condition, deamidation is the slowest. Also, it's highly dependent on temperature. You can see here, 37 degrees deamidate much faster than 4 degrees, which is not surprising. Certainly, there's lots of asparagine are in AAV, and this is the AAV structure sequence. You can see all the asparagine residue are highlighted in red, and some of the hot spot for deamidation, again, in the peptide or no secondary structure are highlighted in different color here. This slide shows the very complex and long steps for AAV generation. And I just want to highlight here, it takes weeks, and the temperature and the pH also change from step to step. For example, at some steps, there's extreme pH, pH 2 and 10 can be applied. Again, if you remember previous slide, these pH can significantly alter the protein structure, at least portion of the protein structure, and can enhance the deamidation or isomerization. So indeed, it's not surprising that several companies and laboratories have discovered that deamidation occurs on AAVs. And this is a paper from Biomarine, and it shows that there are several sites found at deamidation. And also the deamidation correlate to the loss of in vitro potency, shows over here. And the map of the deamidation site on the surface of these residues, and shows here different color. So here is a zoom in view of the deamidation site identified by Biomarine. And one of the deamidation occurs at the VP1 N-terminal residues sites. And as you know, VP1 N-terminal has a lipase-like activity and may be important for export the gene from lysosome, endosome into cytosolic space. So this actually can be very important for the ultimate potency of AAV. And similarly, another group from Jim Wilson's group from UPenn also reported the deamidation of AAV. And this is also highlighted by Nature Biotechnology paper shows over here. And I'll show you a little bit detail on the next slide. So this is really highlight the production of AAV, and the quality control is really important. So here is abstract I get from Jim Wilson's paper, and you can see the extent of deamidation is depend on the vector's age, which is makes sense because this is a time-dependent process. But interestingly, they noticed that the extent of deamidation is independent of the recovery of purification conditions. So that's very interesting. The other thing reported in the paper is Jim Wilson's lab actually selectively mutated some of the residue next to the deamidate asparagine, and then they were to show that they were able to stabilize the AAV and enhance the biological activity. So here is the detailed characterization report from Jim Wilson's lab. You can see they identified several asparagine residue and the deamidate at different degrees. As high can be 99, almost complete deamidate, and some only 1 to 7%. And this is published in the molecular therapy. Next, I want to draw your attention to the potential artifacts during various process, particularly during analysis. So this is something I want to maybe pay more attention to if you have not. So let's even go back to look at the detailed condition report in Jim Wilson's lab. So for tryptic digestion, use trypsin, the AAV was denatured first and reduced and also heated for 90 degrees for 10 minutes, right? So this appears pretty short period of time, but it's much higher temperature. But again, the protein are denatured. And then after this, they did other manipulation. and including alkylation shows over here. And then the digestion with protease trypsin occurs at 37 degrees overnight at a neutral pH. So if we go back, review the propensity of a peptide, right, without secondary structure, asparagine followed by glycine deamidate much quicker with half-life of 24 hours, and histidine and serine, those peptides deamidate about half-life about 10 days. So again, AAV has a well-defined structure, but once it digests into peptide, most likely those peptides won't have higher order structure. So the deamidation will follow this trend. So here is the really the key issue here. If we see a result of deamidate species mixed with asparagine, here I'm only showing the aspartic acid, there's potentially isoaspartic acid. This can mean three possibility, different scenarios. So the first one is if we see, like say 50% of asparagine and 50% deamidate product, it could mean that the original sample has exactly the same amount of asparagine and aspartic acid or isoaspartic acid. So there's no analytical artifact. So again, the results truthfully reflect what's in the sample. However, it could also mean that the sample, original sample, actually have 100% completely asparagine, but during the sample prep and analytical process, some of the asparagine convert to aspartic acid or isoaspartic acid. So this will be an analytical artifact. The other possibility is there are some deamidation events in the original sample, shows over here aspartic acid or isoaspartic acid, but there are more asparagine deamidate during the sample analysis. Again, this will be an analytical artifact. So shows over here is a gray box, right? And ambiguity means the result is not black and white. There's really some uncertainty associated with the analytical process. Again, to show you that the peptide trend, NG for not good, the half-life is about 1 day, not sure, not happy, NH is about 10 days. So you might thought about 10 days half-life is not a big deal, right? But if you actually look at it, if a deamidation half-life is 10 days, so with a 10 hours incubation, for example, tryptic digest or overnight to 24 hours, there could be 2 to 7% of deamidation occurs already. Of course, for the NG sequence, the half-life is 1 day, 24 hours. As short as 0.5 hours, it'll be more than 1% deamidate. Again, if it's 24 hours, there'll be 50% deamidate, and 10 hours be 25% deamidate. So this can be significant. So if you go back to Jim Wilson's paper, you can see that the NH or NS, right? Remember, those half-life of deamidation is about 10 days, and the overnight digestion is somewhere between 10 to 24 hours. So the NH or NS, those two residues, just during the sample prep process, easily can form deamidation species anywhere between 2 to 7%. And you can see what they saw, the result is 7 and 4%. So this raises the question that maybe some of these observed deamidation events are analytical artifact. Right now, we don't know how much of this observed deamidation actually is artifact, how much is a real signal. So what do we do with this ambiguity? So there's good news that we can actually control the deamidation artifacts during sample analysis. So this is one very easy trick we can use is the pH dependent. So it shows here the NG sequence are pH dependent for deamidation at a lower pH. So this is 6 and 5, and the rate is much slower. Okay, so you just remember at neutral pH, half-life is 1 day over here, but at reduced pH, the half-life is much, much longer now. Similarly, for the NH peptide, this is, I'm sorry, the figure is plot opposite. This is actually a longer half-life. In any case, around, again, neutral pH, that the deamidation of NH peptide slow down. At the mildly conditioned, the slowest one occurs around pH 3 to 4. So take advantage of this pH dependent, my student Shan and my collaborator Jared, we develop a method that digests a protein under mildly acidic condition. So on the top is a theoretical mass spectrum of asparagine peptide. And this is based on exenatide, is a GLP analog drug. And the deamidation species will have 1 Dalton mass increase, that shows over here. So, and the normal tryptic or other protein digestion conditions at pH 8, overnight 16 hours, actually they're more than 50% are deamidate. You can see the peak here disappeared. And there's peak here corresponding to the deamidation species accumulate. The good news is if we digest the peptide at mildly acidic condition, here's pH 4.5, even for 16 hours, we don't really see any deamidation. We also see that we can incubate this peptide under pH 4.5 for several days. We don't see very much deamidation at all. So under these typical digestion conditions, we almost completely eliminate deamidation artifact. So what we see is much close representation of the real sample. So the next couple slides is a little bit maybe forward thinking. One of the process is isoaspartic acid. Okay. So we talk about deamidation of asparagine to aspartic acid, isoaspartic acid. So this is one pathway from asparagine from isoaspartic acid. Another related pathway is aspartic acid actually can summarize from isoaspartic acid. Okay. Again, so there's two sources of isoaspartic acid formation. What I want to emphasize here is isoaspartic acid actually form a beta peptide linkage. You can show here is backbone, right? So this nitrogen, alpha, beta, and goes to another amide. Typical amino acid is alpha, amine, alpha, and alpha carbon, and then amide. So this converts alpha peptide to a beta peptide. This generates a huge kink in the backbone and causes significant change of protein conformation. You can show here, the green one is a protein with asparagine residue. Isoaspartic formation shows here, dark blue, significantly change the backbone conformation. Similarly here at another position. So isoaspartic acid have been shown to significantly affect the proteolysis, conformation, as I mentioned, also immunogenicity. So it's very challenging to analyze the formation of aspartic acid, particularly from arrangement for aspartic acid, because these two residues have identical mass, and also they often co-elute under HPLC conditions. Without going into detail, my colleague Barry Karger and we develop a method that uses the advantage of isoaspartic acid resist to proteolysis. We're able to enrich the sample and use a technique called electron transfer dissociation to distinguish this peptide. So another very important question for AAV is we need to think about the whole capsid as one unit, right? Not a single domain of VP1 or VP2 or VP3. So it's really important to analyze the capsid as a whole. So not only intact protein, but as a capsid as a whole. The typical workflow we are doing now is basically digest of peptide. That won't work. So what we really need to do is a way to label, identify the deamidation spot or in the capsid, ideally. And one way we can do that, we can use a chemoenzymatic way to label those proteins. Again, without going into detail, we have developed several ways to label protein. For example, here, we have a protein that we can fluorescent label it. We can also affinity enrich it for subsequent analysis. I'll be happy to share you more details. The last point I want to make, this is based on a series of really nice work by our Greg Flynn and co-workers at Amgen. There's other people who have done this as well, is the concept of biotransformation, or in other words, is what happened of the protein or AAV in vivo. So one example for the antibody field, lots of proteins are stabilized, especially for deamidation at mildly acidic condition. However, once we inject antibodies into our body, that goes to physiological pH and physiological temperature, and then deamidation happens actually pretty fast, especially consider antibody has a pretty long half-life. So I think this would be a very important thing for AAV analysis as well. And there are different approaches that we can use now. For example, we can incubate the protein or AAV with blood, plasma, or serum. So one question is, how do we monitor, right, this process? So there's one way, I think it's a very clever way we can do that, use ONT and water, because deamidation and isomerization all involve water. So if we want to follow a particular step, instead of using normal water, we can use ONT and water. So if some deamidation or isomerization happens that particular step, for example, this is tryptophan digest, then we can see this ONT and water incorporate into the deamidate or isoaspartic acid. And then when we do the mass spec, because ONT and water confer 2 Dalton increase of mass, we can tell what step, whether deamidation or isomerization occurs. So with that, I just want to very quickly summarize my talk. My talk really focused on the analytical artifact. I'll talk about some work people have not get time to work on is isoaspartic acid, intact protein capsid analysis, bioprocessing, how to monitor each step, and how to investigate biotransformation in vivo, maybe do the more pertinent model system. Before I close my talk, I want to just actually run a little bit public announcement for I think, a wonderful program at Northeastern. We call the Industry PhD program, that our full-time employee at the company can also get a PhD at Northeastern. And we have lots of experience, over 20 years experience with this program. And many companies, for example, GSK, Takeda, and other companies listed here has been part of our program. So if you're interested in this program, please contact me. My contact informations are here. And finally, I will be happy to share with you my other actual information papers available, and I'd love to collaborate with you. Again, my contact information is here, and thank you for your attentions.